Glycogenesis. Glycogenesis, it is defined as the synthesis of glycogen from glucose. This glycogenic pathway, or the pathway for the glycogenesis, we can divide it into 4 steps. Number 1 is the synthesis of UDP glucose, and the second step is the synthesis of primers. Number 3 is elongation of chain, and number 4 is formation of branches. We can divide this process, this glycogenesis, into 4 steps. Number 1 is the synthesis of UDP glucose. Number 2 is the synthesis of primers. Number 3 is the elongation of chain, and number 4 is the formation of branches. First of all, what is the synthesis of UDP glucose? In our body, as in the glycolysis, I have explained you that this glycolysis, the first chemical reaction of the glycolysis, is very important, because that reaction is for the trapping of glucose and for the, we can say that, for the different metabolic reactions. Like after the trapping of glucose, we have the glucose in the form of glucose 6-phosphate. That is the first chemical reaction of the glycolysis. This, first of all, this glucose 6-phosphate, it is converted into glucose 1-phosphate, and here the enzyme involved is phosphoglucomutase. Phosphogluco mean glucose phosphate, and mutase mean we are changing the position of phosphate at the glucose molecule. So after this glucose 1-phosphate, it will react with UTP, that is the uridine triphosphate, in the next chemical reaction. As you can see, glucose 1-phosphate plus UTP, and we have UDP glucose plus pyrophosphate. Here the 1-phosphate from the glucose 1-phosphate molecule and the 1-phosphate from the UTP, they are combined and they form the pyrophosphate, and the rest we have only the glucose as well as the UDP, because 1-phosphate molecule is released, and this UDP and this glucose molecule, these both molecules, here the glucose molecule and UDP molecule, they will react with each other and they will form or they condense with each other and they will form the UDP glucose. This UDP glucose molecule is actually the initial molecule to start the glycogenesis. In other words, we can say that this UDP is a carrier, it is a transporter, or it is a, we can say, vehicle which is transporting the glucose to the primers to synthesize the glycogen, to elongate the chains of that glycogen molecule. And this pyrophosphate, this is hydrolyzed into inorganic phosphate. It is hydrolyzed into inorganic phosphate. And what about this UDP glucose? Now, after this first step, after the synthesis of UDP glucose, the second step is the synthesis of primers. What is the primer? Primer is a molecule which is required to act the glycogen synthase molecule, because the key enzyme of this glycogenesis is glycogen synthase. This is a key enzyme, and this enzyme, it only acts on the primers, on the short fragments of glycogen or any sort of primer. It cannot start the polymerization of this glucose molecule into the glycogen. That's why we need a primer. And in this case, there are two possibilities. Number one, a short fragment of glycogen, short fragment of glycogen that will act as a primer, and if that short fragment of glycogen, it is also utilized, or we are also depleted with this short fragment of glycogen, then there will be another primer, that is a protein, that is an enzyme which we name as glycogenin. So here this glycogenin, it is an enzyme and it has an active process which is the autoglycosylation, and as a result of this, like for example here, this is a glycogen molecule. In the glycogen, at the lysine residue, the UDP glucose, it will this carrier or this transporter UDP, it will give off the glucose to the hydroxyl group of this lysine residue. Here we have hydroxyl group, and this glycogen is acting as a primer. This glycogen is acting as a primer, and with the help of its property, which is the autoglycosylation, with that property the UDP will give off its glucose to this glycogen molecule like this. Here we have glucose molecule, and after this autoglycosylation and after the giving of this glucose to the glycogen, we will have the UDP. After this, with this, what we have UDP. This UDP is again converted back into the UTP to carry another molecule of glucose. In what is the pathway for this? This UTP, with the help of nucleoside diphosphate kinase enzyme, the ATP, it will transfer or donate its 1-phosphate to this UDP, and UDP will be converted back into the UTP, like as you can see, UTP with the help of ATP. This ATP, it will donate its transfer its 1-phosphate to the UDP. It is not the UTP, it is UDP, uridine diphosphate. 1-phosphate is transferred to the UDP with the help of enzyme nucleoside diphosphate kinase, and as a result, what we will have? We will have UTP plus ADP. Now this UDP is again ready to carry another molecule of glucose, and after carrying another molecule of glucose, again it will come to the glycogenin, to this primer, and it will give off the glucose molecule to this glycogenin, and as a result, there will be the addition of another molecule of glucose. Then again that UDP will be converted back into the UTP, and again it will carry another molecule of glucose molecule and will give off to this glycogenin molecule. Again this process is repeated, and there will come a point when we will have a short fragment of glucose molecules like this. This is the oxygen. Then we will have a short fragment of glucose molecules. Then the glycogen synthase enzyme, which is a key enzyme, that enzyme will come and will start elongating this chain by the addition of glucose molecules like this to its non-reducing end, like this. And this is the non-reducing end. And what does it mean, non-reducing end? It means the carbonyl carbon or the anomeric carbon that is not available to form the bond, to form the another bond. In other words, we can say that the anomeric carbon that is already being used in the formation of glycosidic bond with the previous glucose molecule. Like over here, suppose, let me explain this with the diagram. Suppose we have here this glucose molecule, like this, and this glucose molecule like this. Here, this is the carbon number 1, 2, 3, 4, 5, and here is carbon number 6. Same with this 1, 2, 3, 4, 5 and 6. This is the carbonyl carbon. The terminal glucose, the carbonyl carbon or the anomeric carbon of this terminal glucose, it is now used in the glycosidic bond, and this anomeric carbon is not available for the formation of new glycosidic bond. So that's why this end is called as non-reducing end. So this was so that's why we call that the non-reducing ends. These are generated as a result of elongation of the chain of glycogen. After that, the next step, the next process is the formation of branches. Here, this was the elongation of chain, and the enzyme involved was glycogen synthase. The next process is the formation of branches. When this glycogen is in the form of in linear form, this is referred as amylose. But as we know that naturally in this glycogen, it is highly branched structure and totally insoluble in the water, and it also has the alpha 1,4 linkages as well as alpha 1,6 linkages, and all the linkages in the linear pattern here, these all linkages are, these all bonds are alpha 1,4, and to make it complex, we need its branching, and with the help of another enzyme which we name as 4,6 transferase enzyme. 4,6 transferase enzyme, which is a branching enzyme. It is also named as alpha 1,4 alpha 1,6 glucoside transferase enzyme. And what this enzyme does, simply, after 11 or 12 residues, in few books you will find 10 to 12 residues, in few books you will find 10 to 11 residues. After 10 or 11 or 10 to 12 residues, it will cut down that chain and link that chain at 6 carbon of the non-reducing end, like this. For example, we name these glucose residues, like we have A, we have B, C, D, E, F, G, H, I, J. In the branching, what will happen? It will simply, it will cut, it will break the alpha 1,4 bond and reform a new bond, that is alpha 1,6 bond. Like in the diagram you can see, and this will happen after 10 to 11 residues. This is glucose, 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 and here glucose, glucose, A, B, C, D, E, F, G, H, I. As you can see that at this point, after each 10 to 11 residues, here on the board I can't show you the 10 to 11 residues, but remember always after 10 to 11 residues, the alpha 1,4 bond that will break, and there will be formation of a new bond, which is the alpha 1,6 bond. As you can see over here, this one, this bond is alpha 1,6 bond, and the rest of the bonds are alpha 1,4. This is 1,4, 1,4 and 1,4. But for the branching, what will happen, that with the help of 4,6 transferase, first of all alpha 1,4 bond will break, and then there will be the formation of a new bond, which is alpha 1,6 linkages or alpha 1,6 glycosidic linkage or bond, and as a result we will have a branch. Now again with the help of glycogen synthase, at this non-reducing end there will be the elongation of the chain, and at this non-reducing end there will be the elongation of the chain, and then again after 10 to 11 residues there will be the cutting down of that chain, and that will be converted into a new branch, and as a result there will be the formation of branches, and at the end we will have a complex structure. So this was all about the glycogenesis, and the main enzyme that glycogen synthase as well as 4,6 transferase. These are the most important enzymes, and the key enzyme for the glycogenesis is the glycogen synthase. Once again I am going to repeat that in the glycogenesis, that is the synthesis of glycogen from the glucose, there are 4 steps: synthesis of UDP glucose, synthesis of primers, elongation of chain and formation of branches. UDP glucose, that is from, as we have glucose 6-phosphate in the cell, that glucose 6-phosphate is converted into glucose 1-phosphate, and that will react with the UTP, and there will be the condensation of condensation after the removal of 2 phosphate molecules, one from UTP and one from glucose 1-phosphate, and we will have UDP glucose, and this UDP glucose is a carrier and it needs a primer to give off its glucose molecule, and there can be 2 primers, the short fragment of glycogen or the glycogenin. If we do not have the short fragment of glycogen, then this glycogenin, which is an enzyme, it will act as a primer, and this glycogenin, with the help of auto glycosylation, it will attach the glucose molecule from the UDP, and then again UDP will be converted back into the UTP and is again ready to carry another molecule of glucose, and then again it will transport another molecule of glucose to the glycogenin. This will go on, and after a short formation of short fragment, glycogen synthase will become active and it will start adding the molecules of glucose, and then there will be the formation of branches, and in the formation of branches what will happen, that after each 10 to 11 residues, 4,6 transferase enzyme, it will come and it will cleave the alpha 1,4 bond, or it will break the alpha 1,4 bond, and it will form a new bond which will be alpha 1,6 linkages, and as a result of this action of this 4,6 transferase, there will be the formation of many branches, and at the end we will have a complex structure of glycogen. So this was all about the glycogenesis.